see the wonderful facilities of this building. Yeah. And so, but then today, yeah, we have what we call double trouble. Yeah. We have two seminars back to back, but they are very exciting. Yeah. So what we're going to start today, we're going to start focusing a lot on One Health. Yeah. And then we're going to move to the interaction between HIV and SARS-CoV-2. Yeah. And what is fantastic, both of our international visitors, they are coming from the USA, and they are here not because us. They are here because they come to the One Health conference. Is that right? And that's something that we found really exciting. Yeah, the amount of people that come to Cape Town for conference, and that we can then ask to come and give us some seminars. So as you are aware, yeah, a few weeks ago, we had a lot of microbiome seminars. They were really excited. We had the ISME, the International Society for Microbial Evolution. Yeah, this week we have the Global One Health Conference. So we really ask them very kindly to be with us. But some of them, especially Julianne, we work for many years, yeah, as part of a program that we have with the University of Washington, the U1, the United World Arboviral Research Network, yeah. And Julianne, she is not a medical doctor, but she's an animal doctor. She's a veterinarian, yeah. Just an epidemiologist, and she's an assistant professor at the Department of Global Health and Epidemiology at University of Washington and Seattle. And today, the first thing that she arrived saying that our weather looked very similar Seattle. And I think that we also have awful winters like Seattle, yeah. But the only difference is that our good weather lasts 8-9 months, and the bad weather 3. And in Seattle, probably is the reverse, yeah. So her research and teaching focus on pastoralist health epidemiology methods from One Health and sustainable theory for modern biomedical approach to One Health. Wow, that looks complicated, but that can be quite the exciting part that we see today. Yeah. In particular, her work applies causal inference methods, spatial mechanisms, models, and network analysis to characterize the influence of political and social force on health at the human-animal environment nexus. So today we're going to have a very holistic presentation on One Health, and then just to invite Julian to do our first talk. Thank you.
Yeah. Okay. Yeah, it's great. All right, great to see all of you, and thank you very much for the introduction, Tulio. I see that you have a lot of presenters recently from University of Wisconsin. So we are the state university and in another state that starts with a W, University of Washington. So as Tulio said, I'm a veterinarian by training, and I actually was fortunate enough in vet school. I thought for a period of time that I wanted to be a zoo vet. So that brought me to Stellenbosch, and I actually spent a month in Stellenbosch in vet school, but eventually did find my way to public health, did my PhD in epidemiology, and now I'm an assistant professor in the departments of global health and epidemiology at the University of Washington. And for those of you who have not had the great fortune so far of being enormously confused by the number of places in the US that bear the name of our first president, I am located in Washington State, which is in the northwestern part of the US and the opposite side of the country from where Eli lives in Washington, D.C. So that's Washington State, sort of a cartoon. Yeah, both Washington, but different Washingtons. We also have Washington State University. We've got George Washington University. We have Washington University of St. Louis, all in different parts of the country. So it's enormously confusing. I am in the northwest corner. It's a very beautiful part of the U.S. These pictures are both from our state, both within a couple hours drive of Seattle. And then on the right is where I live in Seattle. So my talk today is sort of in two sections, and I'm going to try not to give you whiplash because they are a little bit different from each other, but they do connect. The first is really focused on the epi methods that I used, motivated through their application to human African trypanosomiasis. But that's sort of a case study for these methods and how I use them. And then the other is relational One Health, which is the direction that my research has been taking more recently. But the methods that I'm talking about in the first part, I do use to answer questions about relational One Health. So I think those of you in this room are probably pretty familiar with human African trypanosomiasis, but just in case, I'll motivate it briefly. We call it HAT for short, like the thing you wear on your head. And it is a bloodborne protozoal parasite. It's a neglected tropical disease. It's transmitted by the tsetse fly vector. Treatment is quite serious, requires, for the most part, inpatient care, has pretty serious side effects, and there's no vaccine. There are two forms, the chronic form caused by Trypanosoma brucei gambiense, which is in Eastern and Central Africa, and is mostly human-to-human transmission, but there is evidence that pigs may actually be a reservoir for the gambiense form of HAT, which I'll call GHAT for short. And that's really important because we are close to elimination of GHAT. And if we achieve elimination, and there is elimination in humans, but there is an animal reservoir and that isn't dealt with, that could be a source of reemergence like has been observed with guinea worm and dogs. The other form is the acute form caused by Trypanosoma brucei rhodesiense. That's in Eastern and Southern Africa. And that one we know animal reservoirs are very important. But the question is, how important are domestic animal reservoirs versus wild animal reservoirs? Because it's very difficult to control a wild animal reservoir of a disease. As I'm sure you can imagine, that's very, very challenging to do. And because wild animal reservoirs are known to be important, it's widely accepted that it's impossible to eliminate the rhodesiense form. And our question was, first of all, is there epidemiologic evidence that pigs could be a reservoir for the gambiense form of HAT? And then secondly, how important are domestic animals for the rhodesiense form? Could we make inroads towards elimination by focusing on those domestic animals? And in that, we hypothesized there were three pathways for the mechanism by which livestock, which is, that's my term I'm using collectively for cattle and pigs here just to make things easy, but we did everything separately for cattle and pigs. But we're proposing there are three mechanisms by which the presence of cattle and pigs could influence the transmission or the risk of HAT to humans. And the first is the reservoir pathway by which cattle and pigs harbor trypanosomes in their blood, and then that's a source of infection for humans. When a fly bites those animals, they can then bite a human and give the pathogen to them. The second pathway is something called zooprophylaxis. Can I see a show of hands? How many folks have heard of the idea of zooprophylaxis before? Zooprophylaxis. It's kind of zooprophylaxis. Okay, this kind of came out of the malaria. Never heard of it. Okay. So zooprophylaxis is the idea that some vectors show a preference for biting animals, or species preference in general. It could be specific animal species. And that when that occurs, the presence of that animal host that's preferred could effectively deflect bites away from humans. And that was something that was theorized for some malaria vectors, and has also been theorized for human African trypanosomiasis because most tsetse flies do show a strong preference for non-human hosts, in part because humans are much better at killing the flies and mosquitoes that try to bite them than other animals are. And the third pathway is environmental change. And this pathway theorizes that the presence of livestock reduces brush, and brush is a really important habitat for tsetse flies to deposit their larvae and to rest. And there's a lot of evidence that sort of dates from pre-colonial periods that the control of brush is really important for the control of trypanosomiasis. And when either the grazing activity of livestock themselves or changes that people make to the environment to make space for their livestock could actually reduce tsetse fly habitat. So the reservoir pathway I've colored in red because more livestock means more risk to humans. The zooprophylaxis I've colored in blue because more livestock means lower risk. And the environmental change pathway I've also colored in blue because more livestock means less brush and less habitat for the vector. So we set about trying to characterize this relationship in 4 steps. The first was to map cattle and pig density using spatial epi methods. The second was to estimate just the overall effect of livestock density on human African trypanosomiasis risk, where livestock density, what I mean by that is separately the ratio of cattle to humans and separately the ratio of pigs to humans. Yes. Yes, I was going to detail the countries in a minute, but I can spoiler alert, I can tell you right now, it's 4 countries we implemented this research in, all using pre-existing data. So Malawi, which has only the Rhodesiense form, South Sudan, which has the Gambiense, so just the Gambiense form, DR Congo, which just has Gambiense, and Uganda, which has both forms. Uganda is the only country that has both forms. It's where the distribution of the 2 diseases intersect, but within Uganda, they're spatially separate from each other. And then our third step was do an analysis, do mediation analysis for the proportion of that overall effect that is due to the environmental change pathway, isolating the other 2 pathways. And the third was to combine all of 3 of these in the form of a stochastic compartmental model. I'll detail all of these briefly. So the livestock density maps we produced using household survey data, things like DHS and MIX on livestock ownership. And we used an approach called the stochastic partial differential equations approach to Gaussian process modeling, which I will not detail, but folks who want to learn more about it are welcome to come talk to me at the end. The idea behind this is that when you are trying to produce a predicted map of some process that's happening in space, in reality, that process is continuous. Meaning if I move an incrementally small amount from right here to right over there, the value of whatever I'm measuring might have also changed incrementally. You could think about like elevation or something. Any small distance you move, elevation could change slightly, maybe only very slight, but it's not totally flat, right? The world's not totally flat anywhere. And that's an intractable problem computationally. So what you do is rather than model space continuously, you break it down into tiles or chunks. And so this is the discrete approximation to that continuous surface. And what you do with the SPDE approach is you turn space into these triangular tiles that we call a mesh, and you build a spatial model on the intersections of that mesh called the vertices here. And this is the mesh that we overlaid over Malawi. Again, we did this separately for the 4 countries. And then you have a matrix that projects from the intersections of the mesh where you built your model to any random grid that you create, and that's how you create your predicted maps. And our random grid was just under 2 kilometers squared at the equator. So these are just 2 results from Malawi. We did this for every year from 2000 to 2020 for pigs and cattle separately and for all 4 of those countries. There's Malawi on the left for cattle in 2005. We've got cattle in 2010 in Uganda on the right. So next, we combined those maps with data on HAT cases from the WHO to estimate the total effect of livestock density, again, separately for cattle and pigs, on HAT risk in humans. And we had to use a method from causal inference for this called the G formula. And this was required because of something that's happening in the data that I'll explain in a minute. But I also just want to make the case for not being afraid to talk about cause in public health and epidemiology. I think we're taught in our statistics classes that causation is not correlation, and that's absolutely true. But when we're doing hypothesis testing, I think if we're honest with ourselves, we often are interested in cause. Right? Associations are good for prediction, but you can't intervene and improve health if you're only learning about associations. You have to understand what's actually causing health. So just to remind you, the livestock data came from the MAPS and the HAT data came from the WHO. And we had to use the G formula because we had something going on here called treatment confounder feedback, which I'll motivate through livestock and wealth, where wealth is a confounder. So effectively, what's happening is livestock density in a particular location exerts a feedback effect with wealth. And the reason for this is if you have more livestock right now, then you are going to have more wealth in the future because livestock are an asset and they produce wealth for you. And then if you have more wealth, you can invest that in your livestock herd and have more livestock at a future time step. And then that builds more wealth. It's an asset, right? We all understand how wealth accumulates. Wealthy people get wealthier, and livestock ownership is a manifestation of that in some settings. And for the epidemiologists in the room, what this means is that wealth is both a confounder and a mediator, because at some points in time, it exhibits a confounding relationship, the top figure here, with livestock ownership and HAT. And at other times, it exhibits a mediator relationship where it lies on the causal pathway. And the implications of that are we have to adjust for confounders because they bias the thing we're trying to estimate. But if you want to estimate just an overall effect, you cannot adjust for mediators because that induces bias. So you have to adjust and you can't adjust. So you're obviously in a tricky position and you can't just use a regression model. You have to use, there are other methods that you can use, and the one that we chose was the G formula. Now, even if you're trained in mathematics, you might find this slide very difficult to read unless you're actually trained in this particular framework for causal inference, because it uses notation that is specific to that framework and that literature, which I'm not going to explain now because it's complicated and probably won't be interesting to everybody. But again, if you want to dive into it more, I'm happy to talk to you later. The idea here, though, is what you do is you fit a parametric regression model at each time point you've measured to only things that have happened in the past. And that makes sure that you don't accidentally control for a mediator. And then you predict forward using that model, setting livestock density, which was our exposure here, to anything that you define it to be. And you do that for every unit of analysis, which here is a grid cell in those livestock maps, for every year or every time point of your study. And what you get in the end is a distribution for hat cases for the entire study period if every grid cell had been exposed to livestock density at some level that you set. Then you do that whole thing again for a different level of livestock density that you want to compare, and you can take any contrast you wish between those two. Here I did a ratio because it gets us something analogous to a relative risk, which people are used to interpreting as it's quite comfortable to interpret. So our results, just to remind you for the Gambiansi form, the question we were interested in answering is there epidemiologic evidence that pigs are a reservoir for this form? And indeed, in both Uganda and DRC, we found that more pigs had a higher, was associated with a higher risk of HAT. South Sudan, we had some concerns about the validity of the data there. We found some contradictory findings. I'm happy to dive into those later if folks are interested. Rhodesiansi, again, our interest here was in trying to get a sense of how important are domestic animals to the risk of HAT. And in Uganda, we found both cattle and pigs were associated with a higher risk of HAT, so evidence of a reservoir effect, while in Malawi, it was the opposite. We found that more cattle and pigs were associated with a lower HAT risk. And this actually aligns well with other literature on HAT in Malawi, where it mostly occurs in proximity to national parks. And so it is thought that wildlife are really much more important to the epidemiology of HAT in Malawi, while in Uganda, it's more domestic animals. So then we did a mediation analysis. What I just talked to you about was the total effect, the overall effect of livestock density on HAT risk in humans. And now we wanted to tease apart those three pathways that I talked about: zooprophylaxis, reservoir, and environmental change. And we did that by doing an analysis to estimate the portion of the effect that's due to environmental change, isolating the other two. So we used land surface temperature and NDVI, which is a measure of vegetation coverage, as proxies for environmental change. And again, we're trying to estimate what portion of the effect is due to that lower pathway, isolating the top two. And we use the mediational G formula for this. This is a sort of extra extension of the G formula, and you get two causal estimands from this: the natural indirect effect and the natural direct effect. There's no need to interpret these equations. I'll explain them briefly. The natural direct effect is effectively the part that is not mediated, the part that goes straight from exposure to outcome. So you get at this by, when you run your G formula, you change the exposure value. So here that's livestock density, but you hold the mediator value, here that's land surface temperature and vegetation coverage, as fixed. And you get an estimate of the part of the effect that does not go through the mediator. The natural indirect effect is the opposite. And for that analysis, you change the value of the mediator, but you hold the exposure, livestock density here, as fixed, and you get an estimate of the portion of the effect that is goes through the mediator. And we did not find any evidence of mediation across the board. That either means that the environmental change effect is not occurring in these four countries, or that LST and NDVI were just the wrong indicators for it. And then lastly, we tied everything together in a stochastic compartmental model to model the elimination of transmission goal that has been set by the WHO for the Gambiansi form for 2030, is the WHO goal. So we use stochastic compartmental models because GHAT in particular is very close to elimination. And when you get close to elimination, dynamics are characterized by chance events that deterministic models can struggle to capture. The second is that deterministic models use populations that cannot actually be observed to go to zero. So it has to capture elimination of transmission by a proxy. For example, you might say less than 30 cases out of 100,000 in a year is elimination, but you can't actually see it go to zero. With stochastic models, you can not only observe the natural failure of transmission, so transmission going to zero, but you can also model the probability and the distribution of time to elimination. So we had 5 separate stochastic compartmental models, one for each of the 4 countries, plus Uganda had 2 because it has both the Gambiense and the Rhodesiense form. And we set out to answer the question, can GHAT elimination occur by the WHO goal of 2030 under the status quo if pigs are a reservoir? And then for Rhodesiense HAT, our question was, can elimination be achieved through control of domestic animal reservoirs alone? And we looked at 2 One Health interventions here. Trympanoside treatment of animals, which is when you give animals an injectable that eliminates the trympanosome from their blood. And the second is insecticide treatment, where you spray the animal with insecticides that prevents tsetse flies from biting, which not only prevents the animal from getting infected, but it also can kill tsetse flies in proximity to humans. They're kind of like walking vector baits. So this is our model. Again, a separate one for each country and for Rhodesiense and Gambiense HAT. We had a separate model for humans, for vectors, for animal reservoirs. For Gambiense HAT, we had 1 animal reservoir submodel, which was domestic pigs. For Rhodesiense, we had 4 animal submodels: domestic pigs, domestic bovids, so cattle, wild pigs, which we parameterized as warthogs, and then wild bovids, which we parameterized as bushbuck and water buffalo. Yes. Yeah, exactly. Exposure. Yeah, exposed, but not infected. And yeah, recovered. I1, I2, there's 2 stages of infections in humans. And in the first stage is really when they are out and about in their community and can be infectious to other tsetse flies. And then in the second stage of infection in humans, people are usually in the hospital. So they're considered to be removed from the compartment that can expose tsetse flies. They're in the hospital and they can't really be bitten anymore. So yeah, second stage of disease is very, very serious for HAT. People are very, very sick and they are usually hospitalized. That's right. Yeah. Exactly. Yeah, that second sort of removed and in the hospital stage is not, yeah, it doesn't happen for animals. And then we also had non-reservoir animals, which are animals that tsetse flies bite, but they can't become infected with trympanosomes that can pose a risk to humans. With monitor, there's lots of animals that tsetses like to bite. Monitor lizards are a big one that they like to bite. Not really dogs and cats, no. Yeah, I don't know why. Yeah, they don't tend to bite dogs and cats so much. Dogs and cats are probably with those teeth because the tsetse flies hurt when they bite you. Yes, they hurt a lot. You know when a tsetse fly bites you. So I've been told, I've been lucky enough, I've never been bitten. But maybe the sharp teeth of a dog and cat is just not worth the risk to a tsetse fly. So this is the Gambiense form. Under the base case, what you would see here would be a histogram of the year that elimination was achieved. But under the base case, we did not observe elimination at all. This is, that's for Uganda. This is the same model, but this is introducing 50% insecticide coverage of domestic pigs. And we found elimination really, but definitely by 2030, really over the last decade in all of our model runs. So this shows that under the status quo, if pigs are a reservoir, elimination cannot be achieved. But if you introduce some treatment of that pig reservoir, elimination can be achieved on target. And that's effectively what we found across the different countries for Gambiense, is that elimination of transmission was achieved by 2030 under all of the test scenarios, but not the base case. And for Rhodesiense, we did not observe elimination of transmission under any scenario, but interventions did reduce transmission significantly. And for both Gambiense and Rhodesiense, we found that insecticide treatment outperformed trypanoside treatment. If you want to read any of these papers a bit further, the citations are here. Okay, so here's where I give you whiplash by changing gears and talking about relational One Health, and this represents the sort of the evolution of my research, which has really gone from more methods focused to now how do we apply these methods to test hypotheses at One Health hypotheses that also sort of bring in the larger things that are happening in our world. Can I see a show of hands? How many folks have? I think a lot of you have heard of One Health, because I know Tulio has been doing some awesome work now, but has anybody not heard of One Health before? Sorry to put anybody in a spot. Okay, great. So One Health is the idea that human, animal, and environmental health are interconnected. And this is the classic One Health triad. And this has been the sort of symbol for One Health for a long time. It's been, it was very, innovative when it was developed. It's been enormously helpful for describing what One Health is and for motivating One Health. But there's been a lot of criticisms of this conceptualization recently. And I'm going to outline 4 of the main criticisms as I see them. The first is humans are animals, right? Why, we are a type of animal, whether we want to admit it or not. So why are we in a separate circle? The second is who knows what I mean? If I say environmental health, does everybody have a clear understanding of what that means, environmental health? Raise your hand if I say environmental health and you're like, yeah, I know what that is, definitely. No, it's very poorly defined. What do we mean by that? None of us know. Do we mean clean air and clean water? Do we mean biodiversity conservation and ecosystem stewardship? Like, what do I actually mean by this? It's been very poorly defined. And the third is that if you interpret this as a Venn diagram, which I don't think it was ever meant to be, but if you interpret it as a Venn diagram, you could walk away from this saying, look how tiny that yellow intersection is. One Health is not really that important. The fourth, which is not a criticism of this figure, but of the way that One Health has been implemented, is biomedical reductionism. So One Health in its implementation has focused a lot on zoonoses and antimicrobial resistance, and on very anthropocentric conceptualizations of health, by which animals and environments pose risks to humans rather than being health bearers in their own right. And so even though One Health does not, in its theory, impose any sort of hierarchy between humans, animals, and environments, in its implementation, animals and environments have largely been seen as risk factors rather than partners in this world that we share. And another sort of related piece of that is that One Health has focused very tightly on proximal risk factors for those outcomes that we focus on, transmission of zoonoses and antimicrobial resistance. So things like water sanitation and hygiene, food consumption practices, co-housing with animals, things like that. And we've largely ignored the things that happen upstream, the larger forces that shape our world that are becoming an increasing focus in other aspects of epidemiology, including other aspects of infectious disease epi, but are not entering the One Health space really. And by those, I mean the structural and social determinants of health and the global forces that shape socioeconomic inequalities. And if you're thinking, what do socioeconomic inequalities have to do with animals? I have a little sort of vignette to motivate that, to motivate why these things are relevant to animals and to environments. So this is a picture of goats. I think this is not my photo. I think this was somewhere in North Africa. And I'm showing you this picture because I want to convince you that domestic animals throughout the world have also been a subject of colonial domination. So, and other systems of oppression as well. And this is by virtue of who they are kept by. And so during the period of colonization, it was very common that certain animal species or breeds were viewed as dirty or unclean or uncivilized or environmentally destructive, and others were viewed as modern and holistic and nutritious. And what happened as a result of that is that the animal species or breeds that were viewed as unproductive, dirty, environmentally destructive, were often subject to intensive movement restrictions, and sometimes were subject to culls, which is the veterinary term for, strategic slaughter, effectively, while the others receive very heavy investments. And a very clear example of this was in Palestine in the British Mandate period, when the Black Goat Act was passed in the late 40s, which effectively made goats de facto illegal in the Palestinian territories. And that law was only repealed in 2018 when it was recognized that goats are actually really important for reducing the brush that can amplify wildfires in this region. But there are lingering effects of the differential value and investments we've placed in different domestic animals, with one of them being the persistence of brucellosis in some animals and not others. And there are many other examples. And I'm using domestic animals as an example here, but this extends to environments. This extends to wildlife as well. So to address this biomedical reductionism, to encourage more critical theory in One Health, and to better define what we mean by the environment, my colleagues and I developed the Relational One Health framework. Under this framework, humans are subsumed by animals because we are animals, and they are both subsumed by ecosystems. And health is a distribution over all of these health bearers, over the humans, the animals, and the ecosystems that they share. And these health bearers share an environment which is not only biophysical, but is also political, social, cultural, economic, and historical. And they interface with that environment through a reciprocal process called embodiment, which we didn't coin, that comes from Nancy Krieger. And we presented this model coupled with a literature review on other similar One Health frameworks to make sure that we were not sort of duplicating prior efforts. And you can read more about that literature review and our model here. And I'm going to wrap up, I know I only have a couple minutes left, by talking to you about a pilot study we recently finished that sort of informed the development of the Relational One Health framework. They were happening at the same time as each other on land rights, landscape change in One Health. So just to back out and to motivate why we did this a little bit, within One Health, you're going to have to take my word on this. It is accepted as fact that environmental change is the root cause of pandemics. And Eli, you're going to see this talked about a lot in the next couple of days, the World One Health Congress. They're going to be talking about environmental change, landscape change, land use and land cover change over and over and over again. Obviously important feedbacks with climate change. But what we're missing here is what causes landscape change. What causes a forest to be cut down, a grassland to be converted, a rangeland to be converted to pasture, and things like that. And land use change is not a random event. It's not a mystery, and it's not something that happens randomly. Land use change happens because the agents that have control over a piece of land have decided to make a change to it or to not make a change to it. And I want to state clearly that sometimes people have a very good reason for making a landscape change that those of us here in this room might consider harmful or destructive. That's not the focus of my talk. The focus of my talk is zooming in on one particular community's struggle for their land and how that has, what implications that has had for preservation of ecosystems in their territory. So I'm going to take you to Leo's home country of Brazil. I've been working with the Quilombola community of Mata Cavalo, which is in the Midwest, at the intersection of the Pantanal and Cerrado biomes. And this is a complex of 7 community associations corresponding to 7 ancestral trunks, which I'll detail what that is in a second. It's about 11,000 hectares, about 250 households, and they have been struggling for a title to their land since 1999. Territorial recognition was issued in 2007, but they have been unable to receive a title. This is a Quilombola community. Quilombo's communities were established by Quilombola people who were people escaping slavery in Brazil's regional biomes throughout Brazil. A lot of them peacefully cohabited with indigenous communities for hundreds of years and combined their agroforestry knowledge with a knowledge of the indigenous communities that they lived near. And their territorial rights are enshrined in the Brazilian Constitution, but due to a combination, mostly to institutional, structural, and interpersonal racism, which manifests in poverty and violence and social exclusion, most of them have been unable to secure a title to their territory. So only 7% of Quilombos in Brazil have a title to their land. And they are subject to racism in the form of discrimination, interpersonal racism, and violence. And so these pictures at the bottom are meant to show sort of the enormous importance that Quilombos have to the social fabric of Brazil. On the left, this is a picture that we took in Mata Cavalo. This is an installation as part of a workshop that we hosted on Quilombo rights in Brazil. The middle is a graphic novel about Palmares. The Palmares was a colony of about 20,000 Quilombos that successfully fought off invasion of the Portuguese army for about 100 years. So a really incredible story of Palmares there. And on the right is Beatriz Nascimento, who is a well-respected, really famous sort of leader of the Black rights movement in Brazil and was tragically murdered in 1995, but a really incredible writer and theorist. So this is a pilot project where we used a combination of mostly, I work closely with social scientists on this. As you could tell from the first part of my talk, I'm not a social scientist, but we used ethnographic immersion, transect walks, participatory mapping, and questionnaires to characterize the nature of socio-ecological conflict in Matacavallo and how those territorial struggles interface with the health of the Cerrado and Pantanal biomes. And so we were looking to sort of describe and characterize agricultural practices in the territory. They grow a mix of organic foods, herbs, roots, and medicinal products in swidens using a slash and burn approach to agriculture. On the top left, those are castor beans that are harvested for castor oil. On the right is a stump farm where they mix in these multicultural plantings with the native vegetation of the Cerrado, and humidity is very high in these stump farms, supporting the growth of a lot of different croppings. On the left is quite sadly a watermelon. Bottom left is a watermelon rotting on the vine due to El Nino, created some very extreme temperatures in the territory over the last year. And on the bottom right is papaya jam made from the stem of papaya trees. And I want to emphasize that this community is only about 45 minutes from Cuiabá. There's, fantastic modern roads they can take to go into Cuiabá, go to a fancy giant air-conditioned grocery store, but they have maintained these traditional agroecological practices because of the spiritual and cultural importance these practices have to them. They could easily drive and purchase castor bean oil, but they are choosing to produce their own castor bean oil on these traditional methods. So we found that there have been some Western techniques for agriculture incorporated that is necessitated by rural exodus and smaller family sizes. So things like use of tractors and small monocultures in rotation, but there's still very little use of chemical fertilizers and pesticides. We found a strong sense of solidarity with other species in the biomes, which have active roles in the social fabric of the territory, and that survival of the community and survival of the biomes are reciprocal. With regards to territorial struggles and socio-ecological conflict, we found that economic and social vulnerability have been exploited since really the 1950s to expropriate territory, despite the community having a legal right to be there and having retained those legal documents from the late 1800s to today. So they still have possession of these legal documents, but they have not been able to secure a title to their lands. So in the 1950s, this largely happened through non-quilombolas coming into the community, and then hiring quilombolas at exploitative wages, extremely low wages. And then their quilombola workers would get work products, uniforms, food given to them, loaned to them. And then when they were unable to repay those debts were settled by tracts of land being transferred to their employers, basically. And so that was a method of expropriating land. And then this accelerated in the 70s and 80s under the military junta with raids and arson and violence driving people out of the territory and into Cuiabá. So it's the very large exodus. Those expelled families returned in the 90s to 2000s after the redemocratization of Brazil, but they were not given sufficient support for resettlement, leading to conflicts and sales of their land because they had lost a lot of their traditional agroecological knowledge that was needed to sustain themselves and their families in the territory. So they could no longer sustain themselves and they had to sell territory. So these families were twice the victims of structural racism. The first time when they had to leave the territory because of violence and expropriation of their land, and the second time when they were resettled without the proper conditions for that resettlement. And in the 1990s, the Quilombola community was encouraged to accept the settlement of 40 landless farmers in the territory, being told that this would support their territorial claims. But these families practiced a method of agriculture that was more land intensive, and so they gradually began to expropriate land as well. And so on the bottom is a picture demonstrating that. This is a map we produced with participatory mapping. The blue and yellow are the Quilombola communities or Quilombola households. The red is the non-Quilombola. And the green squares or polygons, those are tracts of land that have been registered in Brazil's rural environmental registry by non-quilombolas, which is illegal to do. So those are plots that have been registered illegally as another means to expropriate land from the quilombolas. We found that there are, in the parts of the territory that are still controlled by quilombolas, we found extensive areas of native vegetation that are well preserved. However, at least 5 separate gold mines operate within the territory. This is an area with a lot of gold. And the mining activities were reported by the community to contaminate rivers and reduce soil fertility. So on the left, you have a healthy agroecological plantation. On the right, you have something that used to look like the picture on the left until someone from outside the community took a very large piece of equipment and drove it in to create a gold mine from what used to be an agroecological farm. So our conclusions are that two main mechanisms were used to expropriate land. The employment of Quilombola workers at extremely low wages to establish indebtedness that could only be resolved through the transfer of land. And the second was to leverage predatory legal practices, so that illegal registration of land, to make claims to tracts of land within the territory, and then this ultimately is preventing the community from securing their title. So expropriation is occurring from the outside by agribusinesses and miners, and from within the territory by landless workers. And I think I'm pretty much out of time, so I'll be very quick here. This is a conceptual model developed from our pilot project and from the Relational One Health framework. And we're now, what we've done already is on the left, we've characterized the relationships between land control and ecosystem preservation and their political, historical, social, and economic determinants. And now we're looking to bring in the disease piece. So I motivated this interest in land rights and land use change through pandemics, but pandemic emergence is thankfully quite rare and happens, a lot of times it escapes our notice, but it's quite rare. Arboviruses, by contrast, are not rare, and they're very, very sensitive to ecological conditions. So we are proposing arboviruses as a sort of higher statistical yield test case for this relationship. So in the next stage of our work, we're focusing on quantifying the effect of Quilombola land control on ecological change longitudinally, and using arbovirus or vector surveillance to test hypotheses related to vector abundance and arbovirus exposure risk in different parts of the territory. So these are obviously complex hypotheses. You bring in arboviruses, disease transmission, politics, structural forces. It gets very complicated. So methods development is a big focus of my work, bringing in the first piece of what I was talking to you about. And so I use a mixture of things like spatial epidemiology, ethnography with collaborators, not me doing that, network analyses, vector surveillance, and modeling to test these hypotheses. And that involves collaboration with social scientists. And I think we have in the biomedical sciences the unfortunate tendency to just talk about social sciences as if it's one thing. But there are obviously lots and lots of disciplines within social sciences. So I often work with social scientists from more than one discipline, vector biologists, ecologists, and virologists. And so I want to thank a number of these collaborators. These are just from these two projects. And so the list gets a lot longer when I bring in others. And I think I'm 1 minute over. I don't know if we want to just transition to Eli and folks can ask me questions after, or, yeah, okay, sounds good.
Wow, thank you, Julian. Yeah, fantastic. Yeah. I think let's take a few questions before we change topic. Sorry, I dropped my bag in the middle there. Yeah, I have never seen someone do 90 slides in 45 minutes. Well done.
Oh,
yeah.
I know. Sorry,
but
when I sent slides over.
no, but they were lovely. Yeah. So we have a few questions online, but let's take a few questions here first, please. Okay, I have many questions. Yeah, but before I ask mine, we have two people that have put. We have 50 people online that have put the hand for a long time. Yeah. Tonderai. Yeah, I will just unmute your microphone, and then if you can ask your question. Just a second. Now you have to unmute your microphone. Yeah, and then please. Yeah, go on. Yeah. Just unmute and ask a question. I saw that you almost did.
Oh, I'm sorry. I by mistake. And I'm listening.
Now you need a question. Yeah. Otherwise, we're gonna go to Euchalo. You also have your hands on. I also unmute your. Okay. As they think of the question, we had a few questions in the chat, but as we change here, so So a few questions. How a vet become a mathematician and then move to social science? Because it's a really holistic approach.
Yeah. So I don't do the social sciences work myself. I read a lot of social sciences papers, but I have lots of collaborators who do the social sciences work, and yeah, I find it very interesting. But yeah, my work has jumped around a fair bit.
Yes, and then some question. How do you explain exactly the very description results in both Malawi and Uganda and the DRC? Yeah, I know that in Malawi you try to explain some of the results of the HAT as related to the reserves, but how do you explain, and when you have conflicting results, how do you deal with that?
Yeah. Yeah, so this was a question that obviously reviewers had for us when we published these papers is, Rhodesian NC hat and Uganda, Malawi, it's the same pathogen. Why do you think that there is a positive effect for cattle and pigs in Uganda, but a negative effect in Malawi? And I want to emphasize that when you have different mechanistic pathways that are operating against each other, this was an epidemiological study. It was not focused on, it wasn't as focused on the mechanisms, but more how these two things relate to each other at a population level. So we hypothesize that there are competing mechanisms. There's the reservoir effect, which is positive because more livestock is harmful, and the zooprophylactic effect, which is protective, where more livestock is protective. And because tsetse flies exhibit there's a lot of complexity in their host preferences, and there's a lot of geographic heterogeneity in control measures, you can imagine a situation where biologically cattle and pigs in Malawi and Uganda, if you took cattle from Malawi, cattle from Uganda, put them in a laboratory facility and did experimental testing, that their capacity, their reservoir capacity would be equivalent. There's no difference between the cattle in Malawi and the cattle in Uganda. But what could be happening is the tsetse preference for cattle is different. Cattle versus wildlife is different in Uganda versus Malawi. There's evidence that tsetse fly host preference is a function of the tsetse fly species, but also the animal that particular fly fed on when they first emerged. So if you have tsetse flies in proximity to lots of wildlife, perhaps that population of tsetse flies exerts a strong preference for biting wildlife, versus the ones in areas where there's not much wildlife might show a strong preference for cattle. So there's a difference in the vector population that's going on. They are different species of tsetse flies, so that's also different. And then the availability of hosts is also different. And then the implementation of control strategies is also different as well between Uganda and Malawi. So I think what you could say, oh, it's biased. There was something wrong. Your livestock modeling was wrong. HAT cases in the WHO database are biased. They're underreported. There's lots of explanations for it that are just bias. But and we have answers to those. But if you want to sort of have a more scientific explanation that is not just bias, you could, what it could be is that in both places you have reservoir and zooprophylaxis competing, but in Malawi the zooprophylaxis outweighs the reservoir role, and in Uganda it's the opposite because of the distribution and availability of wildlife versus domestic animal hosts. That was a long answer to Leo.
No. That's a great answer, and we even have a great question here. Yeah, okay. I don't know if I will read the question, but Eruchalo, yeah. If you want to ask a question, I unmute your microphone. So there is suggestion of the use of zebra skin odor smell as a pest control in the domestic habitat. Have you heard about that method?
No.
With a pivotal role in not giving a side effect on the health-wise or the animal or the environment. Yeah. What do you think about that? Yeah.
I guess I would wonder how you manufacture zebra skin odor. I imagine it'd be manufactured. We do use, in the US, wolf urine to get deer. to stop eating your flowers. You can buy it in the store. So there are precedents for using the smell of wildlife to keep things that you want away. That's interesting that zebra skin odor has some pest resistant properties. I wasn't aware of that. I think unintended consequences are always a concern, but I guess it depends on what type of implementation you're doing. If it's really localized, it seems I can't see it. As long as it's harvested ethically and implementation is localized, I can't see a reason not to give it a try and see if it works.
Yeah, and that's sometimes the great things that one learn is a lot of the local knowledge that one have not taught. So and then my last question from you was like, and from the same person, Eruchalo Izuchukwu, sorry for pronouncing wrong. What and how was the estimation of this disease impacted to be associated only with pigs and cattle in the domestic animals? So why did you use only
domestic
pigs
animals
and cattle?
So that reflects both my scientific biases and the data that are available. My research interest is more strongly in livestock than wildlife. So just simple investigator preference there. The other is it's really difficult to get data on the distribution of wildlife. Like the wildlife, they're difficult to intervene on. For domestic animals, we produce these maps based on household surveys where people are asked about their livestock ownership. So DHS, SMICS, the Living Standards Measurement Survey, a lot of them ask about livestock ownership, not because they care about One Health, but because it's a measure of household wealth. So it's really useful for other types of health and demographic questions. So we could leverage that. While wildlife, it's really difficult to get estimates of wildlife numbers. So it's very difficult to model. So that's why we didn't. It was too hard to do. Okay. All right, thank you.
Thank you very much, Julianne. And I know that sometimes that's one of the objectives of this seminar series, is to present you things that you have not taught, because a lot of times that's where the edge of science comes from. Really thinking about different disciplines, and try. And I found really, really exciting. And we are working with Julianne in a few grants. And I think that I'm organizing also with Wes next April. The meeting is gonna be in Stellenbosch, our annual meeting. So we're gonna hear more about that of our PO1 grant that we have at the University of Washington. Now we're gonna change gears. Now we're gonna go for virus persistence and the intersections of pandemics of HIV and SARS-CoV-2. And we have someone else from Washington. But as we learn, the different Washington in the East Coast, that's where kind of Trump live. That's very good. But that is not, hopefully he doesn't live there again. Sorry, I shouldn't be talking about politics. And then I can see shadow face. So it's great to have Eric Boritz. So Dr. Boritz, the head of the Virus Persistence and Dynamic Session at the Vaccine Research Center at the INID in the NIH. So that's where you have the big institutes of the NIH. He's focused on understanding HIV persistence during antiretroviral therapy. And we have here Hert van Zyl. We have multiple people that also work with HIV antiretroviral therapy, and to develop a strategy to target viral reservoirs. His work involves high-throughput method to study infected cells and virus in detail. During the COVID-19 pandemic, he expanded his research to include SARS-CoV-2 and other emerging viruses. And that's something that we also did a lot in South Africa. We pivot a lot for SARS-CoV-2. And one thing that we did, and we also have Tongai. Is Tongai still here? Tongai? Tongai, because we describe many cases of persistence infections with virus evolution. So I hope we see something of that. So thank you, Ellie, and we really welcome you from the other Washington in the other side of the country. Thank you.
Thank you very much for having me here today to speak. Pleasure to be here, and I'm looking forward to talking to you about our lab's work. So I was going to ask a few questions just to make sure I know my audience well. Julia already asked one of them. I know some people in the audience work on HIV. Does anybody here use transcriptomics in their work? If I talk about differential expression, some people know about what that means. Okay, so good. So if anything that I say is not clear, I hope people will shout it out and let me know. Excellent, excellent. Okay. Okay. So my lab is interested in HIV persistence and HIV cure. And this is a slide that I show in the States where HIV has not been top of mind for quite some time, to remind people of the magnitude of a problem that HIV still represents, and one for which we have just a handful or two of reported cures in the literature. And people may be familiar, if you work on HIV, you're probably familiar with how these cures were achieved. They were achieved through hematopoietic stem cell transplantation, which is not remotely safe or scalable enough to represent a real solution to this problem. So looking for something better is an important part of the global approach to the HIV epidemic. So that's not to say that important progress hasn't been made in biomedical research in HIV since the start of the pandemic, since the start, the first reported cases of AIDS back in 1981. I was in grade school when these cases were reported, and it certainly made a big impression on me and part of why I'm doing this work now. So now that I'm approaching middle age, I think it's good to remind the young people of important things that happened before they were born. And one of the most important was the development of combination antiretroviral therapy, where we figured out that combining drugs with different targets within the virus genome allowed us to durably suppress the virus in people who were living with it. And this translated to a real benefit in survival. And this was the first intervention that really showed that more than a decade after the first reported cases. And there was even some question at the time these results came out whether suppressing the virus replication with these small molecule drugs would cure the infection altogether, would eradicate the virus from the individual. So there were modeling studies done using the plasma virus loads, the amount of virus in the blood plasma in the individuals after they started these antiretroviral drug combinations that suggested two distinct phases of plasma virus decay after you started therapy, with the second phase not looking all that long. And this led to an estimate that it might take 2 to 3 years to cure somebody of their infection just by suppressing the virus with these drugs. Now, there were some caveats to this estimate. It assumed a couple of things, that the virus wasn't continuing to replicate somewhere in the body, and it also assumed that there was no unmeasured anatomic reservoir for the virus, because all they were measuring here was copies of the virus genome, cell-free virus genome in the blood. Now, 2 or 3 years later, it became possible to actually do the experiment, and as we all know, unfortunately, the answer was that ART by itself was not a cure, that there was a rebound of virus replication to high levels in essentially everybody who stopped their ART, who stopped their therapy. At the same time this was going on, people were working in the lab looking for reservoirs for the virus in the body. And a couple of important papers published in 1997 showed that the virus could be found in resting memory cells of a type called resting memory CD4 T cells, which were circulating in the blood. And this paper over here showed that if you got the culture conditions just right, you could actually elicit a spreading infection in a test tube, the virus spreading among the donor cells. And then in this study, these infected cells were quantified using a measure called infectious units per million, or IUPM. And the investigators found that the levels of infected cells were quite low. They were about 1 in a million cells. But in this cross-sectional analysis, they looked pretty stable over time. A few years later, it became possible to actually do the experiment better with longitudinal analysis. And it's been done several times now over the last couple of decades, with answers among the 3 studies reporting a half-life remarkably consistent, and leading to the conclusion that we're never going to cure HIV just with antiretroviral therapy alone. We need something more if we want to have a cure. Is this familiar to people? Do people know this story? Okay, good. So that sort of frames our problem in HIV cure research. We have the persistence of the virus during therapy. This is indefinite. And we have this persistent population of low-frequency infected cells within the CD4 T cell compartment, and these cells are stable over time. We don't know that these are the only barrier to curing the infection, but we are pretty sure they're a part of the problem. And so the central biological question that my lab is interested in is, what is the nature of these cells? What are the attributes that allow them to persist? And thus, how might we better target them with new therapies? And an important idea in this area is the idea of the latent reservoir. And have people have heard of this idea? Okay, so this idea holds that infected cells, or at least some of them, have viruses that are in a reversibly quiescent state, and this protects these cells from the virus's toxic effects. Now, this is not just an idea. There are data in the literature to support it, and there are also nice work in cell culture models in test tubes showing molecular blocks to the expression of the virus, the transcription of the virus genome, and then the translation of RNAs produced by the virus, especially in cells that are in a resting state. And this all has led to a cure strategy by which investigators have tried to reverse the latency of the virus, force cells that are infected to express the virus and expose them to killing and perhaps purge the reservoir and lead to cure. So this is sometimes called shock and kill, or kick and kill. And it has now been tested in a series of clinical trials, and they really haven't worked. I don't want to oversimplify things, because how you define the reservoir, how you measure residual virus, is controversial. There's no one right answer of how to do it, but the results have not been very encouraging. And so that's led to a few different pathways forward in the HIV cure research field. There's a sort of practical pathway that says we've cured people with hematopoietic stem cell transplantation. If we give people an HIV-resistant immune system, essentially, maybe there's some technology that we can use to do that, and new gene-based approaches might offer that promise. There are also others who are interested in teaching the immune system to control the virus on its own. That's sometimes called functional cure. And I would put myself in a third category of people who just wants to know better what it is that we're treating. What is the biology of these rare cells that harbor the virus, and can we make progress by learning more about that? So studying these cells is very difficult. I'm showing them here in a cartoon at about 5% of the total body CD4 T cell population, but that's a vast overestimate. It's much less than 1% in most cases of all the cells in the body actually harbor the virus. And then complicating matters, these infected cells do not have distinguishing features on their outsides. They don't have a surface phenotype that separates them from the uninfected cells. So technologies like flow cytometry, which we'd use to ask this question, can't really help us. So you can see what kind of a problem we're in this field. We imagine that there are these rare cells that we can't see that are very important, but we can't understand them because we can't see them, and we don't even really know if they are what we think they are. So people have tried to get around this problem by flipping the problem around, and instead of trying to find the infected cells and then describe them, they've started with an hypothesis about different kinds of cells that exist in the body, and then asked, if we separate cells according to those classifications, do we find differences in how much virus there is, or what kind of virus you find? And I've been a practitioner of this approach too, and there have been a lot of nice studies using this approach, and there have shown some differences, that different kinds of cells have different biologies and they do function in different ways as hosts for the virus. But the somewhat discouraging bottom line is that the virus has been found at low levels in essentially all subsets of cells. And this raises real frightening questions about how distinctive the infected cells are and how difficult might it be to target these cells with new therapies. So what we've needed in the field for quite some time is new technology that can really ask the question we want to ask. If we separate the infected cells and uninfected cells, are they the same or different? How are they different, if so? And how can we use this information? And we need to be able to separate cells that are invisible to our standard technologies, cells that only have that little 10 kb of virus DNA in the 6 billion bases of nuclear DNA. So the project I'm presenting you today was started in 2011, where we thought that part of the solution would be We need to find a way to do high throughput single cell molecular analysis. And the way to do it back then, and I think still the way that it needs to be done at the scale we need to do it, is to use water and oil droplets. All we're trying to do here, these videos can look kind of flashy. All we're doing here is creating millions of single cell reaction containers in which to do molecular biology. And so this seemed to be clearly part of the problem, or part of the solution to the problem. And what we wanted to do in this system was load cells at limiting dilution, break them open, capture their transcriptomes, so the full complement of messenger RNAs that they were expressing, defining their gene expression patterns, detect the virus, and then separate transcriptomes according to the presence or absence of the virus and compare. Now, many of you probably know there are now commercially available systems that do high throughput single cell molecular analysis in droplets. Companies like 10x Genomics, Mission Bio sell systems that are robust and work consistently. But there are challenges here that put it a little bit beyond what we want to do a little bit beyond these commercial systems. Our cell numbers are a bit high. We need to look at millions of cells, not tens of thousands. We have rare cells of interest within those millions. And then, what's really difficult is that we have two different analytes in each cell, and one of them is a tiny stretch of DNA that might be wrapped up tightly in histones in the cell nucleus, not easily accessible for the kinds of things that people use 10x Genomics and Mission Bio machines for. So over a period of 5 or 6 years, we collaborate. So we needed to collaborate. My lab is not an engineering lab. We're kind of conventional biomedical wet lab and sequencing specialists. And so we needed some professional engineers to help us build what we wanted to build. And we found Adam Abate, who is a fluid physicist at University of California, San Francisco, and then his postdoc, Ian Clark, who now has his own laboratory at University of California, Berkeley. And together we built this system called FIND-seq. And so here's what the system does. Cells from study participants are loaded at limiting dilution into water and oil droplets. The aqueous phase here contains a molten agarose that is covalently linked to an oligo-DT capture probe. And what happens when you load cells into the system is that they're lysed and dissolved. The agarose is allowed to cool. It captures the genomic DNA from each cell by size, and the messenger RNA from each cell is captured by hybridization to the capture probe. Then the emulsion can be broken open, everything washed away, the transcriptome of the cell converted to complementary DNA with a reverse transcription step, more washes, another emulsion, HIV detection PCR that generates a fluorescent signal that is then used for sorting of water droplets through oil. So it's pretty fancy stuff that these guys built. And so I'm going to present some results to you where we took purified memory CD4 T cells from people with HIV who had been on long-term ART, and we sorted that material into transcriptomic material, either uninfected HIV DNA negative cells or infected HIV DNA positive cells. And I'm showing you here some videos of these microfluidic machines in action. So these things are happening in about a size on a microscope slide. The device is about this big. There are tubes running in and pumps. Point I'm making here is that these are specialized microfluidic machines built specifically for this biological question. So we used this system to sequence transcriptomes of infected and uninfected cells from a small group of people on long-term ART who were first treated during the chronic phase. And our senior bioinformatician, Prakriti Mudvari, was analyzing the data here. And the first thing that she did was to ask, how different are the infected cell gene expression patterns from the uninfected cells? And so in a global clustering analysis shown here, infected cells are in red, uninfected are in black. And so this heat map makes the point that there was a partial segregation between the gene expression patterns of infected and uninfected cells at the whole transcriptome level. Now, if you look carefully here, you can see that there was also some segregation or some clustering by study participant. So to ask about common themes between participants, the next thing that Prakriti did was to do differential gene expression and pathways enrichment inference, looking for shared pathways that essentially had the same direction across all participants, all 3 participants, from whom we had high quality data. And what Prakriti found was that there were only 6 transcriptomic pathways in the databases that share the same direction in all 3 participants. they were all found to be relatively inhibited in the infected cells compared to the uninfected cells. There was sharing of genes among pathways that I don't really need to go into, but the key point that I want to make here, the thing we discovered was that 2 of the inhibited pathways, death receptor signaling and necroptosis signaling, have to do with the regulation of cell death, and a third, G-alpha 1213, is an anti-cell proliferative pathway. So this was an answer that started to make sense to us, that the cells that are harboring the virus over many years of ART, they might be relatively good at avoiding death signaling, and they might be relatively good at avoiding anti-proliferative signaling. In other words, they self-renew by proliferating, and that's part of what maintains the virus. So this answer started to make sense and started to make us think that this elaborate system that we built might be telling us at least part of the truth. The next thing that Prakriti did analytically was to use a different approach. Instead of doing differential gene expression, to use co-expression network analysis, by which all the genes that were measured in our data set, so there were nearly 18,000, were grouped according to their covariation patterns across samples. And this process generated 28 distinct modules, clusters or modules of genes. And what started to get us even more excited here was that there were only 2 small modules, accounting for less than 1% of all the genes we measured, that were significantly associated with cell infection status, one of them with quite a strong p-value. So the next thing that Prakriti did, we have these gene lists. What do they mean? Do they mean anything? She asked the computer by doing gene ontology, are there biological themes in these 2 groups of gene lists, 2 gene lists that we identified in this way? And she found that there was significant enrichment for specific biological terms. In both of the 2 modules, there was an enrichment for genes related to the regulation of gene expression, the regulation of RNA processing, and certain cell state signatures. So the most exciting part of the study was this part where we boiled the data set down to just our 44 favorite genes, the 44 in these 2 modules that had the strongest signal, and we read the papers. What are these genes? What do they do? What does all this mean? And the best moment for me was what's on this slide, that we found that 8 of the 44 genes that we'd identified in this way were previously shown in biochemical studies to be regulators of HIV gene transcription, including 4 that were negative regulators and were higher in the infected cells, and 4 that were positive regulators that were lower in the infected cells. So what this looked like to us was a host gene expression signature of HIV transcriptional silencing, that the gene expression patterns in these rare cells that have the virus happen to be good at preventing the virus from transcribing its genes. Another thing that we noticed was that among 10 RNA processing related genes in the data, 2 of them that were higher in the infected cells were related to the process of nonsense mediated decay, which is a process by which some messenger RNAs are degraded before they can be exported to the cytoplasm and expressed. And a third of these genes was a stress granule factor. A stress granule is a structure in the cell cytoplasm in which some messenger RNAs can be sequestered, again preventing their translation. So this perhaps evidence of post-transcriptional silencing of the virus in the infected cells. We also found this gene PRRC2A involved in reading certain messenger RNA modifications. This gene has been shown in previous studies to be induced by HIV infection in vitro. There were genes related to the regulation of cell state and fate, including pro-proliferative and anti-death genes, similar to what I showed in the pathways analysis. And then in the remaining genes, there was this group related to the DNA damage response, which for us raised the interesting possibility that some of these gene expression patterns in infected cells might have to do with the cellular response to the integration of the virus or other life cycle steps. So to summarize what we found in this study, which is the more than half of the talk, the first part of the talk, we found it was possible to use a custom microfluidic system to perform whole transcriptome sequence analysis of HIV infected cells. And using this system, we found that infected cells and uninfected cells in their gene expression patterns were different. Some of the differences had to do with long-term cellular persistence and might have to do with just long-lived memory T cells, but some of the signatures of infected cells seem clearly to relate to silencing of the virus. So the findings here link some of these known barriers to cure, these difficult properties of infected reservoirs, to the unique host gene expression patterns of these cells. So the implications here, the first is that these infected cells are selected in vivo, that the infected cells that persist long-term are those that are selected for the ability to do so, including the ability to keep the virus in a silent state. Thus, it makes sense to consider trying to improve this old strategy of shock and kill, which hasn't worked so far. Maybe we haven't sufficiently addressed the different potential mechanisms that can silence the virus, and maybe we might get further benefit from adding agents that can potentiate the natural death of CD4 T cells in the body. But equally important here, that we found these signatures in this first application of a new technology with, the crummy data in your version 1 of your technology that always gets better as you refine it. I think important new discoveries are coming that might open up new possibilities in designing cure strategies. So I'm going to move to the second part of the talk. Any questions on that before I go? Okay, I'll keep going. Okay. So I'll keep going. So HIV doesn't just persist in infected reservoirs. Obviously, it also persists through ongoing replication and sequence evolution. And this is a nice figure from one of the first studies to really look in a really rigorous and detailed way at intra-host evolution of HIV, starting from the time of acquisition and going out a couple of years. And what this figure from this paper is showing you is that there's a progressive accumulation of mutations in the virus genome. And if you look at these colors here, red is non-synonymous and green is synonymous. And down here we have the envelope gene, which encodes the virus spike protein. So you see there's an enrichment for non-synonymous variation in the spike protein of this virus, which is a phenomenon that I'm sure is familiar to you all. And this, of course, this is a challenge. This ability of the virus to evolve its sequence is a challenge in the host, and it also challenges our ability to create interventions that can prevent transmission at a population-wide level. We can't predict what sequence of the virus a person is going to encounter in their life, and so it's hard to know how to vaccinate them, for example. And so at Vaccine Research Center, we want to understand this process. It informs a lot of the interventions that we care about. And so to do this well requires single copy sequencing. And the old school way of doing that is single genome amplification and sequencing, where you do limiting dilution PCR and sequencing of clonal PCR products from your sample material. If you don't do single copy sequencing in chronic HIV infection, you just get schmutz. So you have to do it, and it generates useful information, but it's slow, expensive, and it doesn't make very many sequences for you. So we wanted to have better technology to be able to do this in a deeper way. And I'm going to show you a technology that we have refined. We didn't invent it, and it's not our idea. There's ingenuity here. It's not ours. Other labs were working on this before we were, but we've aggressively applied this technology to other viruses besides HIV, and I'm going to talk about SARS-CoV-2 today. So the way the technology works in the lab, Sung-Hee Ko is a very talented research fellow who has optimized our protocol. Virus genomes are extracted from the sample. They're reverse transcribed with a gene-specific primer that has a sequence barcode or unique molecular identifier, UMI. This material is then PCR amplified and subjected to long-read sequencing, which creates this messy pile of sequence reads full of variation, and much of this variation is technical variation, not real biological variation. We can clean up most of the technical variation with the bioinformatic pipeline that bins reads according to their sequence barcode, and this allows us to get rid of most of the error by consensus formation. There's a little bit of residual error when the reverse transcriptase makes a mistake. We can't fix that with our UMIs, so we have to use inferential methods and drawing cutoffs to try to filter out that stuff. So in the study I'm going to talk about, we had an 8-base random UMI, so 65,000-ish possible sequences. Some of our other studies, we've increased that number. Yeah. And then different UMI designs are possible, and there are good reasons for that sometimes. Right. So if you're loading 65,000 at limiting dilution, you want to probably keep it to a few thousand before you start having UMI collisions by chance. Yeah. So, and this, I should say that this approach to sequencing HIV, I think the first publication on this was over 10 years ago. Ron Swanstrom's group at University of California, of North Carolina, did it using short read sequencing. We need long reads, so we do it with PacBio. And we can get on the order of 1,000 genomes, partial genome sequences per sample. So we developed this for HIV and had been working on it for some years when the pandemic started. And we waffled a little bit about whether we wanted to apply it to SARS-CoV-2 because conventional wisdom was that there was a low mutation rate and the virus wasn't going to evolve. And there were papers in the middle of 2020 saying that we were in good shape, that there was only slow genetic drift except for D614G. There wasn't much going on. And if we had a vaccine today, it would match everybody's virus. Right. Yeah. I was lucky to be at the NIH where I didn't have to apply for funding to do it. I just asked my director, "You think I should do it?" And he said, "Yeah, you should do it." So we did it. And of course, you all know what happened. People here showed the world what was happening, which was that things turned out not to be so simple. And when there were second waves spreading in populations that had some seroprevalence already, all of a sudden there was a new virus there. And there were mutations in the spike protein, and these mutations reduced susceptibility to vaccine elicited and naturally acquired. Well, later there weren't vaccines at this point, or widely applied, but to naturally acquired immune responses. And so the question arose, where do these viruses come from all of a sudden? And that this question became more acute when, again, South Africa told the world about Omicron, which just had such a great evolutionary divergence. How do we account for this very long branch? What was going on back there? We didn't have any clue that this evolution was happening, and all of a sudden we see Omicron. Yeah. I'm not going to answer that question, but I will show you. So I'll talk about this part of the question. Yeah, so it's so funny to have conversations about that, and we're going to go looking for the one out of however many billion people in the world had the virus that was first. That's not feasible. But so one of the hypotheses, as you all have worked on as well, is what role do persistent infections in hosts with compromised immune function play in this? So our initial look at this question was in our first paper using the high throughput single genome sequencing approach. We had access to one participant who'd had a stem cell transplant. This person was admitted to the NIH Clinical Center, wasn't terribly sick, but had the virus for a little while. We had samples in the second and early third weeks of infection. And what was noticed when Sydney Stein and our collaborators' lab quantified the virus RNA and respiratory secretions, it had this jagged pattern where levels of virus declined abruptly between 9 and 13 days after symptom onset, but then increased back up at day 15. And we saw that this was associated with the emergence of sequence variants. And they were interesting variants. There were 4 different haplotypes of the virus at days 11 and 15, all had mutations in the same epitope in the N-terminal domain of the spike gene. And when we look more closely at that epitope, it was at a very exposed region that's been shown to interact with antibodies. When our colleagues mapped the autologous serum antibody binding in this person, they found that the response was relatively late in the course of infection, and it was essentially all focused on this one epitope. So there was a late, weak, and narrow response on one epitope. There were variants in that epitope, and this was associated with a slight delay in the clearance of the virus. So what this looked like to us was the selection, the intra-host selection of escape variants during acute COVID. So we wanted to look more closely at this, and so in order to do that, we wanted to look at HIV positive participants, and we were very fortunate to be able to collaborate with Cheryl Cohen and Van Gottberg at the South African NICD, who had conducted already a couple of studies of people with and without HIV with longitudinal sampling during COVID-19. And so we accessed, in collaboration with them, samples from 48 study participants, notably including 12 who had advanced HIV infection, defined by CD4 T cell counts less than 200 cells per microliter. And in their previous publication, Cheryl and her colleagues had described that it's these people with low CD4 T cell counts that had high levels of the virus in early infection and had prolonged shedding. So what we wanted to know is, how does the spike gene evolve in these unusual cases? And so I'll show you the phylogenetic trees first from the people without HIV. So one tree per person. These trees, as you can see, are pretty boring. Sometimes it's just one leaf and the ancestral root. Sometimes there's a little bit of diversity. So yeah, so let's talk about what's happening over time. So these dots are kind of heat maps of relative levels of the different sequences present on the tree. And what you can see is that for each person in the first sample time point, which is at the left, there's one predominant dot, and then there are a few tiny dots. And the sequence that's the one predominant one at the first day of infection remains predominant until the infection ends. In other words, the virus doesn't get very far evolutionarily in these people. And I should say that this is prospective sampling until clear. So everybody's sampled until the virus is gone. So now, to contrast to this, I'm going to show you what it looks like in people with advanced HIV infection. So you can see this is qualitatively different, where there are dozens of distinct sequences of the spike. The infection is longer in these people, so we have more sample time points, but there's diversity of the virus even at the earliest time points. And then you can see sometimes there's a predominant sequence at the beginning of the infection that completely disappears by the end of the infection, replaced by new sequences. So if you sum all this up, and putting everybody in the study together on a tree really brings home the difference in diversity between people with advanced HIV infection and the other groups. So people whose HIV infection was not advanced looked like people without HIV. That's another important thing to stress. Sometimes people with advanced HIV had over 100 different sequences detected over the course of the infection. Of course, this is just the tip of the iceberg. These are just the variants that we're sure were there in a little swab in part of the nasal mucosa. So how much diversity is there really in the body? It's much greater than we're even able to measure here. A couple of points to make about this diversity. One that I already sort of touched on. Yes, the diversity is dependent on time, but there's also a difference right away at the onset of infection. People with advanced HIV infection have more diversity at the very first sample time point, so something's going on there, and this predicts shedding duration. And the other thing to say is that although part of the difference between people with advanced HIV and the other groups has to be due to more space for the virus to diversify early in the infection, there are also markers of adaptive evolution. So if we look for positive selection, we find multiple sites under positive selection in people with advanced HIV. Only one person in the other groups had any positive selection, and that person might have had advanced HIV too. We just didn't know their CD4 count. So our thinking is the virus simply does not have space to diversify enough, and then the barrier to mutational escape from the immune system is higher in people who have intact immune systems. And then one last thing to say about positive selection and the nature of evolutionary drivers. I don't want to overinterpret our data here, but so I want you to look at these purple curves on top of these two heat maps associated with these two different phylogenetic trees. So in this case, this represents the autologous serum binding to spike. So this is humoral immunity in the blood. And you can see that in this person, after many weeks, this value ticks over the positivity threshold. And this is associated in this person with positive selection on the tree for E484K and a number of other mutations in the receptor binding domain of spike that have been associated with antibody evasion. By contrast, in this person where autologous serum antibody binding to spike is flat, there's positive selection, but it's all outside the RBD. H655Y, P681H, these are not typically thought to be antibody escape, but rather infectivity promoting mutations. So there are potentially different drivers of evolution of the virus in different people where the fitness landscapes may be different. So to summarize this second part of the talk, in people with advanced HIV infection, but not other people with HIV, there was striking intrahost diversity of SARS-CoV-2 spike genes. This was detectable very early in the infection, before the expected onset of adaptive immunity. The patterns of mutations in people with advanced HIV suggested both that there was increased random diversification of the virus, but also adaptive evolution, i.e. selection in these people. And then positive selection was sometimes associated with increases in serum antibody binding to spike, suggesting immune evasion. So with that, I'll wrap up, and just a very quick concluding slide. I talked about here how HIV persists indefinitely within the body of the infected individual in infected cell reservoirs, and these reservoirs have distinctive host gene expression patterns, which may create an opportunity for HIV cure research. In people with advanced HIV infection, many of whom were not receiving appropriate antiretroviral therapy, SARS-CoV-2 can live like a persistent virus, and the evolution of SARS-CoV-2 during these persistent infections can create variants that are a threat not just to the individual, but to everybody else as well. So I don't think it really needs to be said, but really just emphasizing the multiple benefits for people with HIV and people without HIV of better control of the HIV epidemic. So with that, I'll end and acknowledge many different collaborators who made this work possible, groups of members of my group, current and previous, our collaborators in the Engineering Institute, others at Vaccine Research Center, and then our colleagues in Cheryl's group at the NICD up in Johannesburg, as well as the study participants and the funders for the work. With that, I'll end. Thank you very much.
Thank you so much, Ellie. Yeah, that was really, really good. So let's open for questions. I'm sure that we're going to have many questions. Yeah. And normally, traditionally, we like to grill the speaker. I hope you don't mind, and I think that hope people is not shy with two presentations.
Thank you for the presentation. Very interesting. I just wanted to know, did you ever test for recombination between SARS-CoV-2 and HIV for the individuals infected?
Recombination between SARS-CoV-2 and HIV?
Yes. Not sorry. Yeah,
Within.
the recombination within,
Okay.
yes.
Yeah, so great question. So there were 2 cases in our cohort of 47 people. These 2 cases had advanced HIV infection, where we could see that there had been a multiple infection with SARS-CoV-2. So one person had C1, 2, and Delta. I've forgotten what the variants were, but clearly infected with 2 different variants. Another person, we used a cutoff of 5 bases different at the first time point to decide whether there had been a multiple infection, which is a little bit arbitrary, but it seemed good enough. So a few different cases where there were multiple infections, and we could indeed see recombinant haplotypes in those cases. I didn't show that here, but there was recombination.
Oh, okay.
Yeah.
All right. Thank you.
Yeah. Yeah. And so just to round out the thought, there's probably recombination in everybody, but you can't identify it analytically if all the viruses are so similar. But when you get a dual infection, you can see it.
Thank you very much, Gert van Sail. We've done a bit of persistent work in children, but I think this is absolutely fascinating that you presented. I'm wondering about the post-transcriptional silencing as a potential new therapeutic. Are there candidate drugs, and are there any sort of preclinical or phase 1 trials being considered? Because I think this could be hugely exciting for our population. Thanks.
Yeah. No, that part of the reason why our results were so exciting to me is because they seemed true, and they seemed true because people already had that idea, right? People were already working on post-transcriptional HIV silencing. So there have been studies looking at, generally it's repurposing drugs that have this activity that are approved for some other indication. But yes, this work is ongoing, different ways of antagonizing that process. Yeah.
So the HIV cure field, there are questions about where this reservoir is anatomically and also in different cell types, as you mentioned. You show data on CD4 T cells from blood. Are you looking at other cell types and other locations? Do you think the profiles would change?
Right. That's a great question. So yeah, many people in our field write in their paper, the introduction of the paper begins, memory CD4 T cells are the most important barrier to HIV cure, which is unprovable and possibly not true, right? So they're very easy to study. It's easy to draw blood and isolate the CD4 T cells from blood, and that's why we know more about those infected cells than others in the body. So yeah, are CD4 T cells in tissues the same as those in the blood? Well, there are similarities and there are differences. So it's very important for us to study those cells in the tissues. And then are there other cell types, certain myeloid cell types that harbor the virus? There's controversy there, but there are certainly data to support that we need to be looking. Is cell-free virus in immune complexes in lymph nodes part of the problem? Potentially. What's going on in the brain? So there are lots of questions. Some of those questions are not going to be amenable to our approach, but some of them will.
I have a couple of questions for you. Would you mind to put the SARS-CoV-2 3 there? Yeah, the round one that we can see the mutations. Yeah. So by eye, like the most that diversified from the root in a single patient would be what, like 10 or 20 mutations? Yeah.
That's right. From Wuhan. Yeah.
Yeah, from the Wuhan, or from the origin from that patient. Yeah. So how would you explain when you have the big jump of evolutions like Omicron with more than 100 mutations? Yeah.
Yeah. Right.
Yeah. How long would that have to be in an immune suppressed individual?
Yeah. I think I hope I conveyed that our data can't answer that question. Right. 12 seems like a lot in one person, but it's not nearly enough to explain Omicron. So I don't know the answer to that.
Yes.
And I also don't know the magnitude of the total diversity in any one person, just because even though our method is pretty deep, it's still just the tip of the iceberg. So I'm not sure. And there was a nice study in a transplant unit in New York where one patient had some intrahost evolution, and then another patient got that, and there was some intrahost evolution there. So I don't know if you had a serial transmissions in immunocompromised populations, if that would be part of it. I'm not sure.
Yes, because we have one of the we have a couple of papers on Cell on positive selection with Darren Martin. I don't know if you saw it, Sergey. Yeah, and that was a question that does puzzle us. Yeah, despite all the positive selection and kind of directional selection, we couldn't find a way that we could prove or disprove of the immune suppressed individual. Yeah, it is still the most likely theory. Yeah. But one thing that we highlight is even maybe animal reservoir, because we couldn't reject how far the jump of evolution was between some of the strains. Yeah.
Right.
I don't know what you think about that. Yeah.
So the animal reservoir idea, I don't see how to reject that either.
Yeah.
And then, when I talked about these data with people who study RNA viruses, they say, well, yeah, that's a lot of diversity, but probably a lot of that is intra-host variation. That's intra-host fitness, and they're not transmissible. And this is just sequencing, so I don't know which of these variants will transmit well between people. So yeah, there are many unanswered questions.
Yes. Yeah. No. Yes, it's still very puzzling. Yeah. So we have many questions online. Let me see if people would like to ask the question, or if I can read them. Just a second. Let's just find my thing. Yeah. Yeah, here. Okay, you have many claps online. Yeah, first, yeah. So I just want to see, anyone want to ask the question in person, or should I just read the question? So for example, Sabira, would you like to ask your question in person, or should I read for you?